Well, hi. Welcome to Live from One Cancer Place. I'm Erica Hanson Brown, and I'm the creator and founder for One Cancer Place. You might be asking, what is it? Well, it's where the empowered patients come to live. So I want to introduce you to those patients, a few of them anyway, and some of the giants of Cancerland that I have come to know and really, really love and respect. So hosted by Dr. Chris Heery and joined by some of our special patients from One Cancer Place, I welcome you here today.
What we're hoping to do, if we can keep this all together the way we want, is to have sort of Dr. Strickler describe maybe some of the things about his work and how these things develop, and some of the pressures and difficulties that you experience from the side of the physician. Elle to give us the idea of how that looks from the patient side, why we don't see more of that for more patients. And then we can talk a little bit about what are some systematic approaches that might improve that. What can we do as a community that might help improve on those things? So maybe I'll turn it over to Dr. Strickler to introduce himself at this point.
Yeah, thank you. And it's an honor to be invited today. Thank you for having me. I am a medical oncologist, a GI medical oncologist at Duke University, where I'm now associate professor of medicine, also responsible for our GI oncology clinical research portfolio and co-leader of the molecular tumor board here. To start with your first question, Chris, about what is personalized medicine? How does it work? What are the barriers to it? What are some of the successes? I think back to my training, which was not that long ago. So I completed my fellowship training in 2011 here at Duke. And at that time, the management of colorectal cancer was still very much a one-size-fits-all approach. We had a fairly limited number of options: FOLFOX, FOLFIRI, and a few targeted therapies like Avastin and our anti-EGFR therapies, bevacizumab and our anti-EGFR therapies. And one of the frustrations I had is that there were all these breakthroughs going on in the lung cancer world where they were increasingly doing molecular profiling of patients with lung cancer and finding these very actionable alterations in small numbers of people and radically changing their treatment paradigms. And we in the colorectal cancer world were very much stuck in the realm of just giving more chemotherapy. And there was truly an opportunity to apply some of those paradigms to the colon cancer world to help our patients, because it's not just about living longer, it's also about living better, right? Chemotherapy, I hope 300 years from now, we'll look back at the use of chemotherapy and look at it like people look at leeches, for 300 years from now. It works. We do it because it helps people fight their cancer. In some cases, it's very successful, but it's got its limitations and it makes people feel terrible. And one of the exciting things about targeted therapies is that when you have the right drug for the right target, people not only do well, their cancer shrinks, but also their quality of life gets much better too. And that's really what we're striving for. The challenge, of course, is that not everyone has an actionable target in their mutational profile of their tumor. So we need better drugs. We need more targets. And that's an ongoing challenge that we're continuing to work on. But just in the 10 years since I joined faculty, we've made tremendous strides. Now, really what we're talking about here is personalizing the treatment to that patient's individual molecular profile. But there's another aspect of personalized medicine, which is, knowing that patient personally on top of that, right? So no two people are the same. Everybody has their own individual circumstances, their own risk tolerance, their own needs and wants, and we also incorporate that into our decision making. I think it's been an exciting decade for personalized medicine, but we know that there's so much more improvement that's needed. I think this is really just the beginning of this personalized medicine revolution. we do need more drugs. We do need more targets. We need to make it easier for patients to get access to these tests and to these treatments. But the successes we have are so far are impressive. I just wish we had more patients with these kinds of successes in the personalized medicine world. In terms of the barriers, clearly I've mentioned some of the barriers already. We need more targets. We need more active therapies. We're making improvements, but it just feels painfully slow, particularly for those of us and loved ones who are affected with colon cancer. We want those advancements to be happening faster. But on top of that, there are societal pressures and business pressures that also make it harder to give personalized medicine. I think, it's expensive to give personalized medicine. These treatments aren't cheap, and there are pressures for patients to fit into a box, right? And to manage patients along a predefined pathway. And sometimes that makes it hard for doctors to veer outside of the standard path and to do something maybe that isn't FDA approved. And that's something that is an ongoing challenge. I think we in the United States have been blessed in that we historically have given doctors some leeway to do things outside the norm and to manage people along a personalized approach. But there's always that tension, that financial pressure that comes with treating people with therapies off label. And that actually can be a major limiting factor for many patients in certain settings where financial pressures can limit the ability to do personalized treatments. But that's my take on it. I think that that personalized medicine strategies will win the day over time. I think this is the future of oncology care. I do see a future one day where people no longer need cytotoxic chemotherapy. We can focus on targeted therapies and immunotherapies as our primary options for patients. And that, I think that's the vision we need to keep working towards. It's something that I've dedicated my own career to as well.
That's a fantastic overview and lead in. Thank you so much, Dr. Strickler, for those comments. I wanted to turn to Elle now to give kind of maybe her perspective on, what that looked like from the patient point of view when starting out, and how that evolution from sort of the general approach of therapy transitioned to this more personalized approach for you.
Yeah, I mean, just to kind of, run through my story. I was diagnosed in April 2016, so it's been over 5 years now, stage 4. Colorectal, and I had mets to my liver and both lungs. So one of the first pieces of good advice that we got was from my sister-in-law, because we lived in the St. Louis area. And when she found out about my diagnosis, she said, go to Siteman. It was the top, top cancer hospital in the St. Louis area. And I did take her up on that advice. So I went there, and my oncologist at Siteman, Dr. Benjamin Tan, it was his standard procedure to have biomarker testing done right off the bat. So he did the Caris and Guardant 360, and both of those showed that I had the HER2 amplification. At the time, he just kind of noted it and said, it's possible that this might open up some treatment options for you down the road. And that was the extent of, what it meant to him, I guess, at that time.
So what I think, we can point out here is that, for just the other listeners, is that is not typical in most places in the United States. So that right from the beginning, you're already kind of moved into a different category of thinking and planning towards the future, because now something has been identified about your tumor that maybe you're not going to use right now, but at least you already have it on the radar for consideration later, right? Okay. So
Yeah,
I just want to point out these things that tend to be a little different for some patients as you go through the story, because this is an important part of the story. So keep going.
Thank you. Yeah, and please do that, because obviously I just have my own personal experience, and I've learned, things like this along the way. But so yeah, absolutely. Please feel free to interject. So the first thing I did was FOLFOX. I had a near complete response, and then I was part of an online support community where people were saying that the neuropathy can be bad from it, and kind of sharing some results from conferences at the time that were showing 3 to 4 rounds of FOLFOX, pretty much do what they're going to do, and then subsequent rounds don't really do a whole lot more other than make it possible for you to have lasting neuropathy effects. So I asked if we could stop the FOLFOX after 6 rounds. And then we moved to, let me also mention, so part of Dr. Tan's practice was to encourage patients to get a second opinion. And they were going to give us one at the Mayo Clinic.
That's also not typical, right?
Exactly.
So if I think we'll just throw in little bits of advice for patients. If you ever have a doctor who tells you not to get a second opinion, go find a different doctor. That's one of the few hard and fast rules I will say. But a doctor who encourages you to get another opinion is doing so because they have your best interest in mind, not their own ego in mind. So, I would never think less of a doctor who tells you to go get a second opinion. I think that's a fantastic thing, and kudos to your doctor for that. The other thing, just one other point that you mentioned here is stopping the oxaliplatin and FOLFOX early. I just want to point out that this is somewhat still of a debated point in the United States. It's done somewhat more frequently in Europe, at least, from what I hear from people. I obviously don't treat patients in Europe, but I've heard that sort of this earlier stopping of oxaliplatin to limit toxicity, and then just going on Xeloda, for instance, or 5-FU for some period of time, and then coming back to oxaliplatin is sort of more accepted in some practices than others. None of that is sort of outside right now of what people consider sort of normal treatment. There's just variations within those themes of what to do. So I just wanted to point out that wasn't like a decision you made to stop treatment because it wasn't working, or it was for very reasonable sort of evidence-based opinion there. So anyway, so then you're seeking out that second opinion. You went to Mayo.
Yes, yeah, and I think that's a great point too. And in my personal experience, I wasn't really very well supported in that decision when I said, when I expressed my desire to stop after the 6 rounds. That wasn't something that they typically did for patients. They typically just would give up to 12 rounds unless it wasn't tolerated in some way. And I was tolerating it okay. I just was really concerned about the lasting neuropathy. So just to throw that in there, I think it would have been nice for me as a patient, if I had felt more supported in that decision.
And I will say, I think reasonable people can totally disagree on this point. So it's perfectly acceptable that your physician could have looked at it and said, I really think continuing is better for you. I think a reasonable person can reach that conclusion. So I think his suggestion to get a second opinion then makes total sense. Say, okay, well then ask somebody else their opinion on this topic, right? So that's totally reasonable. Anyway, so from there. So then we, looked at the top 3 cancer hospitals, and out of those 3, we really took into consideration which of the 3 locations would we like to travel to more, than the others. Because that's the truth of it, is we're going to be traveling, we didn't know how many times or for how long or anything, but that was definitely one thing we took into consideration. And so out of it was MD Anderson, Mayo Clinic, and Sloan Kettering, and we went with New York. So we asked for the second opinion to be at Sloan Kettering, and he referred us to Dr. Nancy Kemeny, who is world renowned for good reason. And so we met with her.
she thought that, it made sense what we were doing with my treatment. We added Vectibix. So I was on FOLFIRI and Vectibix. And Vectibix happens to be known that it is not very effective or effective at all for HER2 positive patients. And I will say that it was the worst quality of life that I have had of any treatment. So, whether that had much to do with my HER2 amplification, I'm not sure. I'm not sure if my side effects were worse because of being HER2 positive, but they were very bad. And I know a lot of people have, very adverse side effects to the EGFR inhibitors. Then in June of 2017, I asked Dr. Tan what was my prognosis at that point, because I did have progression, after being on those drugs for, I was on Vectibix for over 4 months and was starting to have progression. My CEA was going up. He said that, after failing third line or second line, I think Vectibix is second line, after failing second line treatment, that the median survival is 7 months. And so we were really feeling like we were running out of options because third line treatment is not very effective. He had mentioned Lonsurf, and it just had like a 1 point something success rate or something. So we were really just feeling pretty hopeless at that point. We applied for compassionate access to Herceptin, which is a HER2 targeting drug. But while we were waiting on that to be approved, we checked in with Dr. Kemeny, and they got back to me and said, how soon can you get up here? Dr. Kemeny wants you to bring your paperwork showing that you're HER2 positive. So we got there as fast as we could and did that. And behind the scenes, in the meantime, she had one other HER2 positive patient. And then Dr. Strickler has mentioned that he met Dr. Kemeny at a conference and talked about the HER2 targeting trial that he had, the Mountaineer trial. So when we got there, we were in her office for all of about 5 minutes. And she asked me for the paperwork, and she said, so you have this HER2 amplification. There's a guy at Duke that has what you need. And I'm going to have you go back out to the waiting room, and you will hear from someone to set up an appointment for you with him. And we said, oh, we're planning to go back to, back home tomorrow. And she said, oh, good. So then you can just go there instead. So we had to find arrangements for our kids, our 2 young kids, and figure out a way to get to Duke the next day. But they did contact me and set up an appointment. We ended up taking a train into Greensboro and then going to Duke the next day for an appointment. And Dr. Strickler was our savior.
Story is amazing for so many reasons, but just a few things to point out, because I'm sure this can be a source of frustration for patients. But when doctors go to conferences, we're not all just having fun. This is the kind of thing that comes out of going to conferences: is you meet somebody who's doing something important, you hear about it, you learn about it, and you can make that connection. You actually have their email address, and that's fantastic. Like, that ability to make that connection in a day makes all the difference to somebody like you, right? Mm-hmm. So I'll just point that out. And then the other thing is, having a clinical trial like this open, I think, is pretty remarkable. The timing was remarkable. So I'd love to get Dr. Strickler's sort of background on what led to that trial opening. How long did it take? What was all the background that led to the moment when you met each other that allowed you to go on to this clinical study? Dr. Strickler, did you fill us in on some of that?
I'm happy to tell you about how we got to this trial. So when I started on faculty, I had a lot of interest in liquid biopsies. So taking tubes of blood and running molecular profiles. And I got connected to a company called Guardant Health that was the very first company in this space. And I connected with them because I was interested in a certain target I was looking for in blood, not HER2. And I had this group of patients who were RAS wild type, who had progressed on anti-EGFR therapy. And I was running these molecular profiles through this trial, and I was finding a small but noticeable group of patients we were picking up HER2 amplification in blood. And we didn't know that they were HER2 amplified in their tumor tissue because at the time it was not standard to test that. All we were testing for at that time was KRAS and NRAS. And there are all these treatments out there for HER2 positive breast cancer. And the thinking in the colorectal world up at that time had been, well, you'll never find a patient with HER2 positive disease. There's no point in at developing these drugs for HER2 positive colon cancer patients. And that was kind of the prevailing thinking at that time. If you're not testing for it, you'll never find them. There's no point treating them kind of mentality. Also around that time, there was some data coming out of Italy indicating that a drug called lapatinib, together with trastuzumab, would be active for HER2 positive colorectal cancer patients. So I actually started treating some of these HER2 positive patients with off-label breast cancer drugs, lapatinib and trastuzumab. And one of the frustrations I had about that combination is that the side effects are still pretty significant for lapatinib in particular. You can get rash and diarrhea, and it limits your ability to give active doses of that drug. Right around that time, there was this drug that was just entering the clinic called tucatinib that came out of a small company in Seattle called Cascadian. And I sent them a one-page letter saying, hey, I know your drug is coming into the clinic. It's known to be very well tolerated by patients. It's extremely active against HER2 positive disease. We have these patients, they have no FDA approved options. Let's test this combination of tucatinib and trastuzumab in our HER2 positive patient population. And they, credit to that small company, they got right back to me in an hour and they said, let's talk about it. So it took about an hour, a year for us to launch the trial and open it up. It just so happened that right before I opened the trial, I was at a meeting. We used to have face-to-face meetings not too long ago. Back in the day when we could, talk to colleagues face-to-face while we're waiting at the buffet line or whatever. And I told Dr. Kempany, hey, I'm really excited. I've got this study opening for HER2 positive colorectal cancer. If you ever see a patient with this, it's really rare, reach out to me. We'll get them enrolled. And so Ellie was the very first patient on the planet who with this diagnosis who got this combination. So she's a trailblazer, as you can see. And so it's just kind of extraordinary chain of events for both Ellie and myself that we ended up connecting in this way. And I think the lesson of this story, though, is that so much of this is human to human interactions and human contact and personal networking. There was no computer algorithm that matched us. It was not, there was no, outside forces here. It was just Ellie being proactive, contacting people, asking questions, and then providers behind the scenes, networking, learning what each other are doing, becoming alert and aware of these things. And so much of clinical research really is that. I think that's the part of the takeaway here, that, good luck follows those who are persistent and ask questions, right? And so it was incredibly fortunate for all of us that this happened.
Yeah, it's the saying, chance favors the prepared. But I'm also struck by, sort of your approach to things, because you just told us a story, made it sound relatively simple, but that was probably 5 or 8 years, somewhere in that range of observations, connecting dots, talking to companies, asking for favors, all of that, right? And
absolutely.
I think the question that people wonder about is what makes a doctor have that drive and motivation and still think about it from the patient perspective versus not every doctor can do that, obviously. So how, aside from the sort of the luck that Ellie experienced, I guess, or luck combined with, hard work and effort, how does one identify, the right location to get access to this type of information and ask the right questions to the right person? Because I think in addition to Ellie asking the right questions, she was also asking people who happened to have the information. So that's so how do you have any ideas about how we help more patients bridge that gap?
Well, you're absolutely right, and Ellie happened to establish her care at a clinic that had expertise and knowledge about biomarkers in colon cancer. So she set herself up for success first by connecting herself with a clinic that was willing to go the extra mile. I can tell you that, to your point, not every patient is getting full biomarker testing, even today in 2021, right? And that obviously needs to change. We need to make sure all patients get full biomarker testing. And there are some societal barriers to that happening as well. It's not just the doctor's choice. There are sometimes where it's imposed upon the doctors by third-party payers and other entities. But so I think number one, she set herself up for success there. But there is when you get into the oncology world, I can tell you, we generally know each other. Like I know who's doing colorectal cancer research nationally. We all know each other by our first names. I hope we don't lose this with the COVID era, where we used to interact quite a bit at national conferences, at other meetings. And so much of this is informal communications, right? This is not, this is people standing in line, getting food, talking about what they're working on. I hope we can keep that, but I think also we need to make it not so fortuitous that somebody would land in the right clinic. Like this is something that technology needs to help solve. And we need, honestly, our patient advocacy groups to help be a safety net as well for patients, to make sure that things are happening that need to happen. And I know that's a big part of Erica's mission here. I see her on the line, to make sure that it's not just a matter of lucking out and having the right doctor who happens to know the right doctor, that every patient knows the few questions they need to ask of wherever they're getting seen. So whether they're in one part of the country or another, they're getting the right biomarker testing and they're armed with the right information so they can ask their doctors. Because I think most doctors are very much committed to the health and welfare of their patients. But at the same time, particularly when you get outside of a subspecialty clinic at an academic center, you have to remember that some of these doctors are managing hundreds of different conditions simultaneously, and they may not know about every single target for every single condition they're managing. So I think most doctors actually like it when their patients are asking the right questions, and it forces them to go back and look and say, wait a second, we do have something in our guidelines for this, or we do have some options nationally. And most doctors are pretty good about working with us at academic centers to make sure that if there is something out there, that the patient's getting it.
Yeah. So I want to highlight a few takeaway points for other patients out there who might listen to this, or family members of patients who are, looking for things that can help with their case. So one is, you have the right to ask for this sort of testing at this point. It's available. There is a cost, but oftentimes there are ways to offset that cost in the right setting. So keep asking. Number two is, you probably want, whether it's your full-time physician or at least a physician that is part of your community care team, to at least at some point be in a multidisciplinary clinic that is specific to the disease that you have. Not everyone can see those doctors all the time, but they can see them at major points of their care for critical decision-making points. And that means you can still go to your local doctor, but they're going to execute a plan that came from that multidisciplinary clinic, not just do what they heard about 15 years ago, because the world changes every 3 to 6 months in oncology. And the last one is don't stop asking questions. And I know we're going to keep harping on that, but if you aren't going to be your own advocate, probably no one will. And that's a really important component of this is if you feel like there's not enough new ideas floating around, I suppose there probably aren't, and you should keep asking more questions. So Ellie, I'd like to hear kind of from your perspective then, how did it play out after that? So you made this voyage, you got to Duke. What's the experience been since then, and any other things you could share with patients?
Sure. I mean, Duke is an amazing institution. Just I love the atmosphere, the environment, and everything. And just all the people are, really pleasant. So, I mean, I've been spoiled by that for the last several years. But yeah, just in comparison to the others, the other places I had been, it's just wonderful. The only complaint I have is the toilet paper is really thin. I've told Dr. Strickler that.
It still has the bottom line to consider that. But can you talk a little bit about, then, so if you were meeting a patient for the first time, anything I've missed on takeaway points that you would want to advise somebody who's newly diagnosed, maybe doesn't live in a large metropolitan area with, a great medical center nearby. What are some pieces of advice? Where would you tell them to start?
Oh, I mean, yeah, always the second opinion, and I mean, I guess, that is harder. That's a lot harder for some patients than others to make it to wherever they would get a second opinion. But like you said, pushing for the biomarker testing, because they don't all do it, but it's available to everyone. So, and that if they aren't, willing to get that for you or to give you the second opinion, you've got to go somewhere else.
I wanted to add something to that. I mean, I think doctors actually are very worried about what we call the financial toxicity of what we do. Unfortunately, the system is built in a way where we don't know what the individual patient's burden will be from the things we're doing. And I think that our behavior might change a little bit if it was a little bit more transparent, but it is what it is. And we want the best for our patient in every respect, physically, emotionally, financially. In this particular case, Ellie was flying in regularly from the St. Louis area every 3 weeks for her treatment. And, it is pretty routine for trials to reimburse at least some of the cost of travel. But I think the way that this was an investigator-initiated trial that I wrote and was funded by this company, small company in Seattle. And I, was worried about her financial burden. So I sent a note back to the company and I said, look, we need to help our patients who are coming from a pretty significant distance every 3 weeks. And they agreed to pay a substantial amount of the travel costs. And once again, it was just a matter of understanding the financial burden, and then we can advocate. And the company was willing to pay for it because they knew that this is a rare biomarker and they're looking to get the study enrolled, and they did not want the financial burden of travel to limit people from being able to participate. And I think that I just give kudos to Cascadian, which was bought by CGen, and CGen, when they took over the trial, and CGen is supporting this program, but when they took over the trial, they kept that financial support in place so that the burden of traveling to the site would not limit a person's ability to participate. And so, kudos to them for keeping that going, because that's really the most patient-friendly thing we could do.
Yeah, no, that's fantastic. I'd also point out that, initiating an investigator-initiated trial is a lot of work, and having one actually work on the first patient must have been the most rewarding thing I can say from my own perspective. That's never been the case. So congratulations on a good idea that worked out. So Renee actually put a question in the chat that I think is a great one to share. And Dr. Strickler, maybe you could comment on this, which is that, she had a tumor biopsy or a tissue biopsy from the primary, and then later had another site of disease. And the question is, how often should you repeat testing to look for actionable targets at different sites of disease? And maybe you could talk a little bit about tumor heterogeneity a bit there too.
Yeah, that is such a great question, and something that we need to get out there for education for both doctors and patients. So tumors are not static, and they change. They can evolve under the pressure of treatment, and this is something that is known to occur with any kind of personalized medicine strategy. Is the personalized medicine strategy works great as long as the tumor maintains its genetic profile. But that tumor is fighting back the only way it knows how, and that is to evolve a way around that personalized medicine strategy. So in terms of how often to think about, repeat profiling of that tumor, I would say if the patient's on a targeted therapy and there are signs of progression, either a rising CEA or a new spot on a scan, it is my practice to profile that cancer again. So specifically, that can be done with a liquid biopsy through a cell-free DNA assay, or you can physically take that tumor out and then send it off for tissue profiling through one of the diagnostic companies that we have available. And what we learned from that is we, number one, try to understand if the target is still there or if it's been lost, or if there's some other mutation or driver that has been acquired by the tumor that allows it to escape that personalized strategy. And so that should be considered at those junctures where you think maybe that personalized strategy is not working any longer.
Yeah, and I think maybe I'll just add one more part of this question, because I think it's a challenging topic sometimes, which is there is sometimes an underlying assumption that a true driver of disease will be present sort of throughout, right? Honestly, I don't think we know enough about the evolutionary pressure that occurs within a given patient to be able to say that definitively, and to therefore rule out the question of whether you should resample. So I don't know, Dr. Strickler, if you have any comment on that, but I think it's just too hard to make that assessment. Is that kind of something you think too, or would you think about it slightly differently?
No, I mean, I think there are certain mutations. I have been doing this long enough and seen enough of these profiles. There are certain mutations that are kind of foundational to that tumor, and I know will always be present whether we do a liquid biopsy or a tumor tissue biopsy. And then there are others that can come and go. And so part of that is just having that experience of knowing the patterns of evolution and being able to anticipate them and being prepared for them as a doctor. And then being able to work with the patient, when those new targets emerge or when the tumor evolves in a way that changes what we need to do next.
Yeah.
I think it's this is an area where there's a lot of research going on and we're still learning quite a bit. And yet another reason why it's good to connect with physicians who can provide that expertise to understand how to interpret those reports and when it's time to get a new report.
Yeah. Julie asked about the Mountaineer study as an example of taking a therapeutic option from one tumor type to another, just for sort of broader awareness. There's also a term used frequently called basket trials, where a target is leveraged across different tumor types. The question, Julie, let me know if I didn't get your question fully, but it sounds like your question is, should patients be sort of looking at their profile and then looking for basket trials into which they might fall, and what's the method by which they could identify this? Can you comment on that a bit?
Yeah, I mean, I think this is something that's been going on increasingly over the past 5 years, where whenever there's a successful therapeutic strategy with an actionable drug, actionable target in one cancer, where the next question is, okay, where else is this target? Who else can we help with this? And that can be done through a basket type design. And that's where a basket is where you have maybe 5 different targets, and whether it's HER2 or BRAF or PIK3CA. And basically, if you match up a target, you go into the basket where your tumor matches. So that's what they mean by basket trial. And those have been done with some success, particularly in the HER2 space, for a few years now. And then occasionally there will be a company that develops a new drug with a new target, and they say, hey, it works for this cancer, let's try another one as well. And one of the things that I'm encouraged by is that the FDA is increasingly allowing us to break down these disease barriers in how we look at applying these drugs. So there have been what we call these tumor agnostic approvals over the last couple of years with a few extremely rare targets. Things like NTRK is an example of that. And so the FDA is actually making some changes that makes it easier for us to break down these artificial barriers, because while it's not always true, frequently if it's actionable for one disease, it's probably actionable for another. But there are some examples where the disease matters too. And so it's also important to understand, well, you can't just treat a BRAF mutated colon cancer the same way you treat a BRAF mutated melanoma, and here's why. There are examples where you can't just take away that disease designation. The disease does matter, but in many cases it doesn't.
Mm-hmm. Yeah, I know this topic can be quite confusing for patients. So I just wanted to add one, maybe a little bit more on the tail end of what you just said, which is that there can be genes that are either mutated or expressed in a given tumor cell, but the cell from which that cell derived may actually impact how important that pathway is in that given disease. And what I hope everyone takes away is that this is another situation where having someone who both understands sort of personalized therapy and your specific disease is really helpful, right? So if you went to see Dr. Strickler, for instance, or somebody else who does looks at the world the way he does, he'd be able to apply his own personal experience of that given pathway in that given disease better than someone who may be interested in that pathway, but say in a different disease, or may have a different perspective on it. So I think that's just another little nugget there that we can all take away, is that there's not a short-term view of cancer converting over to just, it is completely agnostic to the organ from which it derived. I know we all hope for that someday, but we're not quite there yet. And so we still need to put them in context of kind of where they came from, at least for the time being. So still really important to see somebody who has experience in the disease as it's titled today. So Ellie, I also wanted to hear a little bit from you about how you're now using what you've learned to map out what your future looks like.
Mhm.
I know that's kind of a tougher topic, but are you thinking about this in terms of, all right, what can I do to prepare for if this therapy isn't controlling the disease anymore? How are you thinking about that at this point?
Um, to be completely honest, I have such an amazing doctor in Dr. Strickler that it's like I am able to just, trust him, and he, we, every time I have scans, we go through it, and like every so often we will discuss, what other options are kind of out there, and like plan A, B, C kind of thing. And so, I mean, there's nothing I need to do besides that right now.
No, but that having sort of a series of potential plans is, I don't think that's typical. I don't think that's
Exactly.
the typical
No.
patient experience.
No,
So,
not at all.
Yeah.
So
So
yeah,
can you talk about that, like about how that feels from your perspective, to be able to know that here's a list of things from which I'm considering. You don't need to know all the details, but they're on my target list. I mean, can you just talk about sort of how that makes you, just your mindset? How does that affect you in sort of your
yeah,
quality of life?
yeah. I mean, just to know that, that like my how I feel about how often we're doing scans, like is taken into account. I mean, I do defer to, what Dr. Strickler thinks, for the most part, but yeah, it's like my input is always taken into account. And so, like I said, just, as often as I want to, really, we can discuss what are we thinking. And so that's just such a nice thing to be able to rely on, and to know that every time we do have that conversation, it's not just, here's what's next. It's like, here's how things are, evolving. And so, yeah,
Yeah.
I mean,
That's fantastic. I think my biggest concern coming out of this call is that Dr. Strickler is going to have a lot of new patient self-referrals to his clinic. So sorry about that.
There are also, I will point out, there are many wonderful medical oncologists nationally who take great care of patients, and you should feel confident that, your medical oncologist is more than likely fantastic as well. So that said, but one of the exciting things for Ellie is that even since I met her, her options have proliferated. She has so many more options therapeutically now than she did in 2016 when she was diagnosed, 2017 when she came over for the trial. And that's really exciting too. The science is evolving rapidly too. So, we had a smaller list of options back then, but we've got a huge list of options now. And it can be pretty overwhelming for a patient to say, wait a second, I've got this embarrassment of riches now. I can't decide which thing to do next. And, that actually can be pretty overwhelming for people too. To have like 3 or 4 great options and not know which one is the best. So, we work that through, and we help to prioritize those options and go through the if-then statements as scans come up. And, it's a partnership. And I think that's what you're hearing from Ellie, is that we really work together to make sure that we get her the best treatment possible and still allow her to live her life and, do all the things she wants with her family and her kids, and not be limited by things that we're doing as well.
Yeah, I think that's a fantastic point to end on. That, and Ellie, you referenced this a couple of times, but part of the relationship between the physician and the patient is also for the physician to understand what matters to the patient enough to be able to tailor timing of visits and what kind of treatment and how much you travel and all of that stuff, right? So I think that's a really great message to also share with patients that you just did, Ellie, that you can say this is what matters to me, right? I mean, it's more than reasonable to say, I'd really prefer to do this because this is what I like. You hear this language around the battle with cancer a lot, but for a patient, it's also about, well, I don't want that to be the thing that defines the rest of my life potentially, right? So I think that's a really important point, and I'm glad we got to touch on that as we're running out of time here. I want to thank you, Dr. Strickler, for for all the work you did to create a trial that helped Ellie to be here looking healthy. I think that's just like the biggest reward you could ask for, but also for sharing your insights with us today. I hope this is helpful for the people who listen in. And I would recommend, for patients to maybe, rewind, go back at 2x speed now and try to capture some of these little nuggets that we heard throughout, because there were a lot of great pieces of advice. But any last comments or thoughts you want to share, Dr. Strickland?
Well, I think that it's incredibly emotionally, physically draining to be a patient. And I recognize that, and I see the toll it takes on people and all the sacrifices people make as they go through their treatments. And, I want people to know that their doctors do care. We are, highly motivated to find better treatments, less toxic treatments, get people more time, better quality time, all these things. And when we get together at conferences and talk about things, that's what we talk about, is how we can get the next breakthroughs for our patients. And that's something that really motivates us. And I think, as hard as it can be sometimes to be an oncologist, to be an oncologist at a time where we're making these breakthroughs is so exciting. And, it's such a positive thing. And also I wanted to thank all of you who are involved in this meeting today, who are helping to get the message out there for patients that, there are biomarkers that can be tested. There are things you can do to help yourself so that you do have a better journey. And so, thank you also to everyone for having me here. And I wanted to say thank you for having this event.
And now we're going to have our separate session, which is a patient conversation with Dr. Heery moderating as well. But to kind of debrief what we heard and anything, so we can continue to kind of ask questions and engage around some of the topics that brought up that with Dr. Strickler.
Renee, sorry, I didn't see your question until just now, but I can try to answer that. Yeah, exclusions in clinical trials are real. So her question was, for someone just viewing, was this question that comes up a lot around patients being excluded from clinical trials based on prior therapy experience. So for instance, if you have had a checkpoint inhibitor, and then there's a combination trial with a checkpoint inhibitor, oftentimes that combination will exclude the use of prior checkpoint inhibitor. There's not really a good answer to this question. I'll tell you the two perspectives on it, and then at least you can understand why this happens. The two perspectives are these. One is any patient who has already had their disease progress while they were on a checkpoint inhibitor, has already had their disease sort of demonstrate the ability to overcome immune mechanisms of killing, right? So therefore, if you take 100 patients in that population, you might have decreased the likelihood of success by some percentage, right? So instead of maybe 35% of patients responding to that combination therapy. Now you're going to get to 25 or 20% if the combination really matters. And so from a company strategic standpoint, what they're trying to do is control for expectations in the marketplace. And those expectations are ridiculous, right? Most people that invest in companies like this do not understand that 25% response rate after having had a checkpoint inhibitor, for instance, and having disease progression is an excellent outcome. All they compare it to in their minds is what is an objective response rate that I expect to see that predicts long-term likelihood of that drug working in a randomized controlled trial. And that number, for better or worse, tends to be about 35% in people's minds, right? And I'm just telling you, what are the factors that lead to these decisions? They have more to do with money than they do with actually holding fast to trying to help patients in the near term. However, and I say this because I have to live in this world too, if you don't live up to those expectations, you're going to run out of money before you can even run the pivotal trial. And therefore, a drug that might be a very good drug will never make it into the hands of thousands of other patients. So you're making the choice about what do you do now to help patients that might go into trials versus what do you do now that might help hundreds or thousands or tens of thousands of patients later. And that's a really tough choice. I can tell you when I was an investigator, I pushed as hard as I could to get as many patients on the trial as I could. Now that I see it from the other perspective, I say, it's not that simple, because if I do that, not only am I going to undermine what might happen to any given patient on that study, I'm also going to undermine the drug. So I don't have the right answer. I don't think anyone does, but I'll just tell you it's a complicated problem. And typically, if something looks like it really, really works, even in the patients who have had prior checkpoint inhibitor or whatever, then eventually it will make its way to the group of patients who have had prior experience with that therapy and now in combination later. But it does usually take another, couple years to get there, which can be frustrating for someone who needs it right now. So,
Yeah.
Is there some of it there too, where some of these drugs, and I don't know exactly, but since it's really a question, is some of these drugs, they don't know what the long-term therapeutic benefit is of it. And so if you've been on it before, you don't necessarily know if that's still having an impact, or is that just really not that big of a deal?
Yeah, it absolutely can be. I mean, I think take pembrolizumab, a drug that a lot of people know now because of its relevance in, MMRD, colorectal cancer, but in lots of other cancers. The problem is when you progress, when your disease grows while you're on that therapy actually can stay in your body for about 6 months or so. And it's also possible that biologically, the effects of that drug could have long-lasting effects, even after it's washed out completely from your body. So that is a hard thing to solve for, especially if what you're trying to prove is what we call a mechanism of action. If you're trying to prove that a drug works a particular way, you really don't want any confounding variable on board there. And so again, early in development of a drug, you try to keep it as clean as you can to understand how does it work and in whom does it work. And then later you'll expand that more and more and more once you know the answers to those questions. But yeah, these are, unfortunately, while we've increased the number of therapeutic options, we've done so through increasing complexity of biology. And it is very difficult to sort out that complicated biology if you do a mishmash of lots of different patient populations and lots of different therapies, which doesn't make any individual patient feel any better. I totally understand that, but it is the reality.
I guess I have a question for Ellie. How have your side effects been with this trial?
Yeah, I mean, that was something we didn't even really talk a whole lot about, but that's huge, because like I said, the EGFR inhibitor was the worst quality of life I had. I went from that to starting the Mountaineer trial, and I've had it's full quality of life. And that's still the case, even though I had to come off the trial this past August or April. I'm on Herceptin and Perjeta now, which is really similar, targeted therapy. So it's still, I would still describe it as full quality of life. It's like I get a bump on my face every now and then. And so I know that's probably from the drug because I haven't had that happen the last few years, but it's so minimal.
Why'd you come off the trial, Ellie?
I did have progression, so I had never been eligible for surgery. But then when I finally did start to have some progression on the Mountaineer trial, it was near the primary site, and they reevaluated me for surgery and deemed me eligible just because the trial was controlling the Mets so well. So they were able to operate. So I had the colon resection last February, 2020, and a temporary ileostomy that was reversed in June. And then I had more growth in the liver. I didn't have to come off the trial, at that point. I came off the drugs, the treatment for surgery, but then I was still on the Mountaineer after that. And then there was some growth in the liver. So I had a liver resection this past February, and it still didn't have to come off the trial right away because we had addressed the growth, but then there was still it started to come back after that. So I did come off the trial at that point. So, but I mean, I've been good since switching to the Herceptin Perjeta. Dr. Strickler described it as throwing it a curveball.
Yeah, that topic, I'm sorry we didn't get there, of why would a patient with a given target be able to tolerate a therapy better than, if they don't have a lot of that target on their tumor, is a really interesting one. And some of you may be aware that, for instance, with EGFR targeting monoclonal antibodies, one of the things that people tend to say is a predictor of whether patients will have long-lasting benefit is whether they develop a face rash with that therapy early on, and then you can kind of scale back dosing a bit. The reason for that, I'm going to give you a very, very high-level overview, but it has to do with pharmacology, which is sort of the study of how drugs distribute through the body, and then what their effect is. So there are kind of two main points within pharmacology. There's pharmacokinetics, which is where the drug goes and how long it lasts, and all of that. And then there's pharmacodynamics, which is what is the effect of the drug when it gets to where it's supposed to get to. And in the case of a targeted therapy, the presence or absence of the target actually affects both of those things. That's a little bit of a confusing idea. So I'm going to just pause for a second. I'm going to say it again in a little bit different way, which is, think about it this way. If you have a bunch of things that you want to hit, and you have heat-seeking missiles to go after them, your heat-seeking missile, absent the target, is just a missile, right? So any destruction that it can cause, absent the heat that it's going after, is going to happen. But if you have that heat or that target, and it's going to localize right to where it's supposed to, then the toxicity in the rest of the body may diminish as a result, which is why you can kind of dial in the therapeutic, what we call the therapeutic window of a drug, when you've gone past the toxic level, to the toxic level a little bit, but you see the tumor shrinking, you can kind of narrow that dose in on just hitting the target more than the rest of that normally expressed target on normal cells, right? Mm-hmm. So the case of HER2, similarly, HER2 is on normal tissue. But with the therapies we have available, you can use dose so that they will preferentially go after the cells that have too much of it, like in this case, the cancer cells, right? Mm-hmm. So again, you can kind of dial in the dose to for the utility. And I mean, it's not like personalizing that dose in this case. It's that people have already worked out what's sort of the best range of doses that'll hit the target on an overly expressing cancer cell versus not on normal cells. So I hope that helps and it didn't add to the confusion, but this is one of the things that is sort of the magic of chemotherapy, because chemotherapy is just poison, right? So the magic of it is that over many years, people have figured out a way to give you just enough poison to kill your cancer cells, but not you.
Yeah.
And that gets better and better and better the more targeted the therapy is.
So a follow-up on that, because most clinical trials, right, they look to kind of the highest tolerable dose, right? So you have a drug like, a lot of them, but Stivarga or Longsurf, which are known to be pretty tough treatments to take. And the nuance of that dose, right? In terms of like, if you have really bad side effects at the highest dose, and I know the redose study on Stivarga kind of tried to address this, and it's like, okay, ratcheting it up, but then your oncologist, right? Most oncologists don't necessarily know that. What they know is what's been studied, right? Because how would anybody know beyond that? But an oncologist who's not like focused on like understanding that like perfect little place, or maybe it's the pharmacist, I don't know how that works, where you say, look, this dose was the highest tolerable dose for the population, right, in the study, but for you, it's too high. But how do oncologists figure out where to go from there, because it's like somewhere between 0 and that dose is the right amount, but like what do they do?
Yeah, I mean, oftentimes that's years and years and years of trial and error in clinical trials to know what those ranges that are possible could be, and experience. I mean, it's a combination of those two things. In current drug development, like in the last 10 years or so, maybe 15, there's been much more energy and effort placed on understanding the biomarkers that predict outcomes of a therapy and toxicity of a therapy. So, as we evolve through our therapeutic landscape, what we're seeing now is that most of the time when a drug is approved, it'll come with, this is the standard dose, but you can increase the dose or decrease the dose depending on these factors. And that's usually in the package label, right? So in fact, there are even chemotherapy regimens in lymphoma, which is considered a highly curable set of diseases. But in one subset of lymphoma, that the therapy is actually designed so that you increase the therapy till you hit a toxicity level. And the reason is the toxicity that you're trying to hit is damaging normal blood cells. And we know that in that particular regimen, in that particular disease, that when you hit that threshold, you are really destroying the cancer cells at an extremely high rate, right? That's not true for every cancer, but you can see what I mean. There are ways that you can sort of follow the effect of the drugs up until the threshold you want, and then either back off or find a level ground. And in colon cancer is a good example of one where we've had the drug 5-FU, for 30 plus years, whatever it is, right? As a backbone of colorectal cancer treatment. And really, not until recently has anybody even been interested in understanding, should we change the dose based on some parameter of the patient? And it turns out, if you understand the mechanism through which 5-FU works, there are actually some relatively simple biologic correlates that you can measure in the peripheral blood that tell you, in this particular person, am I overdosing or underdosing to the effect that I want? Again, those things are still in clinical trials being evaluated, but the question you're asking is like, why isn't that the standard? And it's because some of these drugs were developed before these methods even existed to sort of evaluate these things fully. So hopefully that will be the future, though, that we will think about doses as a range around which within which we need to work and then understand which part of that range fits that patient better.
It's like, and when you bring up 5FU, it's like the bolus of 5FU. And, most oncologists will say, or, CRC specialists will say, like, don't do the bolus, just get the long term, but it's not in the guidelines. And, when I was thinking about it, I was like, it's because whose incentive is it to test that? Like what,
Nobody.
like who, like the drug company, it doesn't help them. It doesn't help, it's kind of like, well, it's easier to leave it than it is to change it when it causes such issues for so many patients,
That's such a good point, but I guess I'll put it to you this way. There's only one version of 5FU where the dosing is still being evaluated in an ongoing fashion, right? And that's the one that's still on patent. Yeah. So if you want to know what drives this type of work, it is definitely, is there a financial reward in it? And certainly having a therapy that people can tolerate longer and benefits patients is financially rewarding for companies. Mm-hmm. But for standard 5FU, that's a generic drug, right? So, but Xeloda, there's still money to be made by capturing more of the patients to use Xeloda versus infusional 5FU. So, and I don't mean to make it sound quite so crass, but incentives drive behavior in the real world. So, and then I have to... Like
biomarkers question, right? Because Ellie, in your case, like your initial oncologist probably at the time didn't really have much to say about her too, because there wasn't anything to say necessarily. And had, and we talked about the confluence of events to get you to the trial and be the first patient on that trial. There's all these, like you get these biomarker results and you say, okay, these 2 are actionable. These 10 you have, but they're not really actionable. So like, anybody's opinion, who kind of has used their biomarkers, like in terms of how often do you check on those things? How do you check on those things? Like what's the even Renee, to your point on, getting updated biomarkers, but even on the ones, but they're not actionable yet. Like, what do we do? I don't know, Ellie, like what?
When I had my liver resection in February, they did another biopsy and a biomarker test, and it did note like a new amplification, MET, a MET amplification, and so Dr. Strickler said, that's what he suspects the growth was from, and so we're just kind of like keeping that in the back of our minds, and in case it is something that we could try, because I think there are some targeted therapy drugs for it. So
there are, yeah, and I think that's, but the way you're phrasing the question, Julie, is right, which is it totally true that no one had conceived of using HER2 targeted therapy even when it was found on your first panel? Well, no, I mean, Dr. Strickler was already really working on it then. Right. I mean, so again, if you had happened to have been in his clinic,
right,
he might have said, "Oh man, I gotta bend over backwards. I gotta do everything in my power to get this study open immediately." Right.
Or go off label, like.
Yeah, or go off label. Right. So
right.
I think that's really the harder challenge, is that when you see something like that and there's no context and nothing's been published or presented yet, how does anyone know what to do with it? I think that's a real challenge. And I think it's not a challenge we're going to solve on this call or in the near future.
or maybe ever.
Right.
But it does support this idea of going to visit the people that think about this all the time, at least once in your treatment decision-making, right? Because you just need somebody who thinks about this stuff and talks to people about this stuff all the time to be able to get those insights. And I want to protect Dr. Strickler again. Don't everyone call Dr. Strickler for that. There are people all around the country that do stuff like this. And maybe what we could do is attach, when this gets uploaded, maybe Dr. Strickler could give us a list of like 10 or 15 people that do similar work to him around the country, so we can diffuse this out around the U.S.
In listening, Ellie, I'm so happy for you. I'm just so happy for you. But everybody needs to be their own best advocate, but as someone who's just new to the cancer world and you're still in shock from your diagnosis, how in the world do you figure out what questions to answer, who to ask, or what questions to ask, who to ask them to? It's just overwhelming.
I just read about a study that I think I posted in One Cancer Place where they did pathologist consults with patients. So that idea of, having an expert on the pathology, basically doing a consult with them so that like a patient consult, so that you're able to understand from somebody like that what it all means to some extent, which I think is actually a really powerful idea because, then it arms you with information to be able to share with your oncologist. Your oncologist gets the information. I mean, it's kind of like, any other specialist consult. So I don't know. What do you think about that, Chris? That idea?
Hang on. I think it's the right concept. I think it's hard to implement, right? I mean, most pathologists don't have a lot of clinical insight. I mean, some do, some are amazing, but most don't. Most are, in most places, are going to be covering a lot of different diseases and being asked to wear a lot of different hats. You do find at some big academic institutions a pathologist who is actively involved in the multidisciplinary team, who knows the targets, all that stuff. But that's not typical. And, honestly, most pathologists chose pathology so they wouldn't have to talk to patients. So I don't know. I think there's something to it, but I think it's better if it's through the translation of somebody like Dr. Strickler, to be honest.
Yeah.
Like, a pathologist that they can work with and feel comfortable with, and then they translate it for the patient. And I'm really sorry, but I have a meeting I have to get to. I really appreciate you all having me back, and look forward to the next one. Thanks again.
First Wednesday in December.
I'm gonna mark it off.
Hello.
All right. Have a great day.
Everybody.
Thank you, Dr. Harry.
Thank you.
Thanks, Chris, so much. Thank you, Sejan.
Yes. Thank you, everybody.
Thanks, everybody, for showing up.
Thank you.
Thank you, Ellie.
Thank you,
Yeah.
Lovely in blue.
This is our movie room. We painted blue.
That's good. It's like the sky when you're watching a movie.
Have a good day.
Bye,
Thank you.
everybody.
Bye.
Bye.